Hello, everyone. Um, Yesterday we discussed something about Acanthamoeba. Uh, that lecture actually was divided in two parts. So here today we are going to discuss the third part. So that's why I'm going to, include some glimpses of the previous lectures in order to make a continuity in this lecture for the audience. So we discussed about Acanthamoeba that doesn't require any host. There's no vector is not required for transmission. It's not well adapted to parasitism. The classification of this particular parasite. Then we discussed the current interests in this particular parasite. Uh, Apart from this, that it produces very serious infections in human, its role in the ecosystem and its role is a host or reservoir for microbial pathogens causing different types of diseases is also very important, and that is creating a lot of interest in this particular parasite. And it's also used as model organism for motility studies and a number of other studies that we will discuss today in this particular lecture. Then I talked about the pathogenic species and different conditions that it causes. For instance, I talked about the granulomatous amoebic encephalitis, which is a very fatal condition, inflammation of the brain, which is caused by this particular parasite. And we talked about infections of the lungs and skin that it causes in immunocompromised individuals. Those individuals who have some kind of weaker immunity due to transplantation, grafting of some tissues, or due to some diseases like HIV, or due to taking some steroidal drugs, or some other conditions like cancers or heart disease. If people have these kind of conditions, then of course they can develop this condition as a result of this parasitic infection. In healthy individuals, it causes chronic amoebic keratitis. We discussed about that. Then I discussed about the evolutionary history of this particular parasite, and we came to know that it probably would have emerged, in between periods that are specified for the emergence of plants and fungi. So that was the evolutionary time period. The discovery was made in 1930s, and it was actually discovered in a culture of yeast, which later was confirmed as an amoeba or a species of amoeba, which was named as Acanthamoeba. And now it has been associated with these two conditions as we discussed, but this association was made long before, in 1960s and 70s, with ADE and keratitis. If you look at the biology of this particular parasite, we discussed this that it has got spikes, just like the coronavirus. It also has spikes, but these are different in nature. But the name acanth means spiked amoeba. So Acanthamoeba means spiked amoeba. There are different vacuoles for specialized functions, ranging from osmotic regulation to glycogen storage vacuoles, digestive vacuoles. There are lysosomes containing hydrolytic enzymes. The nucleus is just like any nucleus in the eukaryotic cells. It's a double-layered membrane protected by a double-layered membrane. Normally there is one, but there can be many. Some studies suggest that there can be many of them. One-sixth of the tropocyte, it would take about that much volume inside the tropocyte. The plasma membrane, again, is a typical plasma membrane, just as you would have seen in case of any other eukaryotic cells with proteins, phospholipids, sterols, etc. And all these things are required for different functions like reception of signals or transportation, or some would act like channel proteins, other things. So these the functions of this plasma membrane is also similar to the functions of other eukaryotic cells. Mitochondria are large in numbers. They are chloroplast as well, and you know that what is their function. Genomic size is variable, 41 to 45 MB, and the genotypes, there are 17 known genotypes named from T1 to T15. And T4 genotype in more than 90% of the cases is involved in this, keratitis. And T1 and T4 genotypes, they cause infections in different organs. For instance, T1 only infects the brain and cause inflammation of the brain. But T4 genotype, it can cause inflammation of the skin, inflammation of the brain, or inflammation of the lungs. So it can infect different organs, of course. The habitat is diverse habitat. It's found everywhere. It's ubiquitous in distribution. If you look at the morphology, two major forms, trophozoites and cyst, it is found in these two forms. There are more than 20 species of this particular parasites, and they are divided into three groups based on the morphology of the cyst, the size and the size of the cyst, or in its morphology, the appearance of the cyst. You can divide this parasite into these three major groups. So the trophozoite is exostate, it's feeding stage, and uh, the parasite normally would exist in this particular form at 30 degrees Celsius or at neutral pH, and when the nutrients are available, then you can find the parasite in this particular shape as you would see on the screen. It's feed on single-celled organisms, including many different types of bacteria that you would know in today's lecture, Mycobacterium avium, for instance. It's a bacteriovore. So the cyst, it changes into the cyst form when the conditions are not favorable. It's a double-layered membrane. You can see uh, in this particular slide. It's an outer cyst which is, ringled, and there is an inner cyst wall, and there are different, organelles there. This is the nucleus and other granules inside, vacuoles and granules inside the cyst that you can see. So these are interchangeable forms, the cyst and the trophozoites based on conditions. Distribution in the environments we talked about, it's everywhere, in hot springs, in water lakes, in hospital settings, on vegetables, and you name it, almost everywhere that you can imagine. This parasite can survive everywhere. So you can see that how prevalent this particular. This is the pathogenesis. Its infective stage is the trophozoite. The cyst would convert into trophozoites, and the trophozoites then would get into your bodies by two different means. With the help of aerosols, the aerosols can either go into your respiratory tract through the lungs and then would join the blood and reach your brain. On the other hand, aerosol can also be, uh, you know, uh, taken up directly by through your nose, you know, and they can also reach the brain through that particular route. And the skin penetration is another route through which you can uh, be infected with the trophozoites, and from the skin it goes into the blood, and then again uh, uh, the destination is the same. And in healthy individual that causes this condition, you can see the difference between these two eyes. It's caused by Acanthamoeba keratitis, which is um, this particular amoeba that we are discussing about. And it is prevalent in people who use contact lenses, or who would receive some kind of trauma to their eyes. So their eyes are injured and they just wash it with contaminated water, then there are chances that they would get this particular disease. We have discussed this. Then we discussed about the immunology of this particular infection, that the first line of defense is provided by the tear fluid and the blinking movement of your eye. These are protective moments, the first line of defense against any pathogens. In the cornea, the outer layer that we discussed, avascular. If it is avascular, it means it would not be protected like any other vascular structures would be protected, because the blood supply is not there, so you cannot expect a cellular or humoral immunity here for protection. So what is here? The tear film, which is composed of aqueous, mucus, and oily layers, three different layers, and the aqueous layers particularly contains these and antimicrobial lysozymes, lactoferrin, and secretory immunoglobulins. And on the other side, in the inner side, conjunctiva is a highly vascular structure, and this immunoglobulin A producing plasma cells are there, T lymphocytes are there, natural killer cells and macrophages are there. So it means that inside the conjunctiva, cellular and humoral responses for clearance of this particular parasite would be, initiated. And inside on the cornea, at the cornea, there is no such response as cellular or humoral response. Only these endomicrobial lysozymes, lactoferrin, and secretory immunoglobulins have to protect your cornea. And the blinking moment of the eye, which has to spread all these things over on the surface of cornea to protect the eye. So in order to proceed with the infection, we discussed is that Acanthamoeba must remain at the corneal surface and invade into the stromal tissue. And the tear film, in context, why contact lenses lens were prone to this condition, because the tear film it is kind of under stress due to that particular lens. It becomes thinner, and the inflammatory mediators in tears are their level is altered there. So that's why. And the second reason would be occlusion of the atmospheric oxygen uh, and thinning of the basal layer. So that already have been discussed. So once in the stroma, Acanthamoeba secrete proteolytic enzymes. We discussed this, and would bring about stromal degradation, and it will lead to infiltration of the macrophages. Macrophages would secrete cytokines, which would attract neutrophils, neutrophil infiltration. So there will be a kind of cell-mediated immunity in action here. And this, in turn, will lead to modulation of the B and T cell responses, which, of course, would lead to this antibody based immunity or humoral responses would be generated for the clearance of this particular parasite. And now, this is today's lecture. It starts from the growth cycle, that how this particular parasite actually is growing. So you can describe either in the form of phases: lag phase, exponential phase, stationary phase, cell death, and encystation. Or alternatively, you can divide the phases into two different steps. Like you can describe it as interphase. The interphase period is divided into the following phases again. A G1 stage. In the G1 phase, various molecules including enzymes are synthesized as the cells prepare for the next phase of growth. Some of the cells start to divide in this particular phase. S is the DNA synthesis and replication takes place during the S phase. G2, during this phase, DNA synthesized in the S phase contributes to high protein synthesis to produce microtubules. About 90% of cell division also takes place during this particular phase. The microtubules produced during the G2 phase are important for mitosis, or cell division that occurs in the M phase. M phase or mitotic phase. So in the M phase, nuclear division occurs in the M phase. chromosomes are separated to form two daughter nuclei in preparation of cell division. While nuclear division as well as cytoplasmic division occurs during this phase, the M phase only accounts for about 10% of cell division in the growth cycle. So the growth cycle is also described in phases after it divides by binary fission. As I told you, there is a lake phase. The lake phase, if you describe it, this is the first phase in which the cells take time to adapt to their environment. It is also during this stage that the Acanthamoeba cells start maturing. Some of the molecules synthesized as the cells mature include enzymes and RNAs, etc. So that's the lake phase that we discussed. Then it is followed by the exponential phase with favorable conditions available. Favorable conditions mean nutrients and temperature, etc. The number of cells conditions, to increase in the exponential phase. If the conditions are favorable, then the number of cells automatically they would increase. The cells continue to double until nutrients are depleted. In waste, toxin material increases. So depletion of the nutrients and the buildup of toxins lead to the next phase in the cycle. The next phase in the cycle is the stationary phase. The stationary phase is the result of higher number of cells from the exponential phase, depletion of nutrients and increase of toxins. These conditions results in reduced rates of growth. And the next phase is cell death and incestation, and as a result of depleted nutrients and high levels of toxins, the cell starts to die or form cysts to ensure their survival. So cyst formation allows the cells to survive in various extreme conditions by remaining dormant until environmental conditions improve. So cell death and incestation, these would take place at the end when these conditions would become adverse for the parasite to um, survive. So that's why it would undergo incestation, just like the cyst formation that we discussed in the last lectures. That why cyst formation would take place in case of these uh, amoebas. Okay, so what is its role in the ecosystem? It's very important to understand this, that the protists, like amoeba, flagellates, and ciliates, they have two major roles in the ecosystem, and that is influencing the structure of the microbial communities which are present there in the ecosystem and enhancing nutrient recycling. Okay, so how does these protists, amoebas, flagellates, and ciliates are doing this particular job? You know that a lot of work this of decomposition of the organic matter is carried out by the primary uh, decomposers, and the primary decomposers are always bacteria which are doing this particular job. But these bacteria have a problem. When these bacteria they do this process of decomposition of organic matter, lots of nutrients they remain trapped in bacteria, so they are not good at releasing the nutrients trapped in them. So that's a problem. So it means that the nutrients would not be recycled with the same efficiency as if with which they are taking them in. It's a problem for the environment. So amoebas are very helpful, for particularly Acanthamoeba are very helpful in this particular conditions because they consume 60% of bacteria. The free-living amoebas are feeding on bacteria. They are bacterivores. And 60% of these primary decomposers are decomposed by the secondary decomposers, which are these amoebas. And these amoebas are just, digesting them, and, they are just helping the environment in a sense that they are providing these minerals trapped in the bacterial systems back to the environment. Okay, they are giving it back to the environment by decomposition of the bacteria. So 60% of them is decomposed by them, and in doing so, it is influencing the microbial communities in uh, the environment is well. Many different types of microbial communities, bacterial and fungal communities, are affected. Many viruses, bacteria, fungi, etc., they are living inside these acanthamoebas, and they also thrive. They replicate there inside acanthamoebas, some of the wild species, and you know. So it means that these are very important for maintaining balance of other microbes in the environment. So, and that balance is again required for, is important with respect to survival of species in this. And that's why it's called the Trojan horse of the microbial world. Because this particular parasite, it harbors endosymbionts, which may include viruses, yeast, protists, and bacteria. It has got this endosymbiotic relationship with these different types of pathogens, including viruses, bacteria, and protists, and they are living there, as I told you. For instance, they are hosting the largest known viruses. For instance, the Mimi virus, 1.2 million base pair is the genome of this particular virus, which is too big in size. Or the megavirus, so it's another category of big viruses, are the pandoviruses, which were discovered in 2013 and have the largest, viral genome so far discovered in the, in case of viruses. If apart from these megaviruses or mimiviruses, Coxsackie viruses, adenoviruses, polioviruses, ecoviruses, enteroviruses, vesicular stomatitis viruses, and a lot of viruses that are of pathogenic nature. They are living inside these Acanthamoeba species, which is quite striking. Can you imagine that coronavirus would be living in a similar parasitic species, or parasitic species would host the coronavirus, and it would just live there, and sometimes, you know, it would come in contact with the human species again, even if this pandemic situation just goes away. So there are chances, because these parasites are also acting as hosts for different types of species. Now, there are not only the viral species, there are lots of other bacterial species. Some of them you would have heard their names of. For instance, Toxoplasma gondii, which is a very famous parasite actually living there inside these amoebas species. In itself, Toxoplasma gondii is a pathogenic parasite. And it's living here inside Acanthamoebas, and would come out and infect its species, other species from there. Bacterial species, you would find many different types of bacterial species like the Staphylococcus. Whatever you can see out of this list, whatever catches your eye is important, like Listeria monocytogenes cause listeriosis. Staphylococcus aureus or methicillin-resistant Staphylococcus aureus, which is a big problem in terms of control. Antibiotics are not working in many cases. Mycobacterium tuberculosis, which again is a big problem. It's living here in Acanthamoeba. Pasteurella multicida is living here inside Acanthamoeba. Salmonella typhimurium, Shigella, and other species, pathogenic species causing diseases. Campylobacter jejuni, the phyla pneumonia, Bacillus cereus, all these pathogenic species, including E. coli, 0157, neuropathogenic coli, K1, neuropathogenic E. coli, all these are living inside this particular Acanthamoeba species. So you can just imagine, the importance of these Acanthamoebas in, in the environment, and for other species survival are, uh, acting as they are acting as reservoirs for other bacterial and fungal and viral species. So you can imagine how important their role is in the environment and, uh, you know, uh, for human beings, because uh, okay. So the next is as model organism. It has been used as a model organisms since long. Acanthamoeba has long been studied as model eukaryotic cell with special emphasis on the actin cytoskeleton-based mortality. So it has it moves quite faster. It's about 0.8 meter per second. So its movement is quite faster, and it is using the same kind of machinery. It's almost the same as any other mammalian cell. It has the actin filaments, and it is the cytoskeleton and the weights, movement is typical of other the movement of other organisms, other uh, um, uh, you know if somebody wants to study the details of this moment at the molecular level, then they can use Acanthamoeba as a model organism. It is used to understand the molecular biology of cell motility. At deeper level, you can understand what kind of different molecules are involved in this process of mortality. For instance, what kind of different proteins, what kind of neural proteins or immunogenic proteins they are involved in this particular process. All this can be accomplished by using model organisms. They are also used to study cancer cell dormancy or to understand the process of incestation and depth. So you can just use this particular organisms. It's also used for studying the evolution of certain G proteins or GPCRs, and also used to study the function of the voltage gated calcium channels, because the voltage gated calcium channels inside these acanthamoebas are similar to the one found in human beings. So that's why if you use some blockers of these channels, you can carry out such studies using acanthamoeba. And Alzheimer's disease, of course, Alzheimer's is a very important disease. It's also studied at molecular level in this particular model. So lots of different compounds and their functions, proteins and their functions have been described by using this particular model, Acanthamoeba. So that ends today's lecture about Acanthamoeba. You can subscribe to the channel and for more lectures. And if you have any questions, you can just ask the questions, and I would be very happy to answer your questions. Thank you very much.